Can you still hear me, Shaunak?
Yes, I can. Okay, I think it's right on time. Hello, everyone. Warm welcome to the Biologically Speaking webinar series. I am Shaunak.
Can you still hear me, Shaunak?
Yes, I can. I can. Okay, I think it's right on time. Hello, everyone. Okay, sorry for that. I'm Shaunak, postdoctoral fellow at the National Cancer Institute, NIH, in the USA, and also co-founder of this platform, Biologically Speaking, which we started exactly 2 years back in the middle of pandemic with a group of PhD students and postdoctoral fellows. I'm delighted to say that we are entering our 3rd year of this initiative, and we are thankful to our sponsors who has helped us along the way, specifically Bioswatch India, American Society for Cell Biology, and more recently, the Women in Bio Initiative, the Chicago chapter in the United States, has helped us to achieve this long journey where we host scientific talks in the form of webinar, and all of our previous talks are archived on our YouTube channel, where we are currently live streaming today's session as well. So if you'd like to be a part of our initiative, please write to us via our website at www.biologicallyspeaking.com. Around middle of last year, we initiated a new series called Crosstalk, partnering with Dr. Mohit Jolly from the Indian Institute of Science, Bangalore. And together we discuss behind the scenes of collaborations where we talk to scientists and their trainees who got benefited from the collaborations, how to initiate and sustain a productive collaboration in the scientific field. And today is the first session of Crosstalk for this year, and I'm absolutely delighted to have our panelists who are distinguished scientists in the field and have been collaborating for a long period of time. So before I introduce our panelists, a couple of housekeeping things to remind. Please keep your microphone muted and videos turned off during the session. And since this is more of a discussion, we encourage our audience to engage and interact. And you can post your questions for our speaker using the chat box, and I will moderate them at the end of today's session. With that, our first panelist is Professor Ambarish Ghosh, a renowned physicist and a currently professor at the Indian Institute of Science, Bangalore. He earned his PhD in physics from Brown University in Providence, Rhode Island, followed by a postdoctoral fellowship at the Harvard University. He returned to India to start his independent laboratory at IISc Bangalore in 2009, where he is currently appointed as a full professor. And the lab is focused in generating these nanoswimmers for understanding some of these biological phenomena. Our next panelist is Professor Ramray Bhatt, currently an associate professor at the IISc Bangalore. He earned his medical degree from the University of Calcutta, followed by a PhD from the New York Medical College. Professor Bhatt did his postdoctoral work at the Berkeley Labs and then moved back to India to start his laboratory at the Indian Institute of Science, Bangalore. Professor Bhatt have been collaborating on multiple fronts, specifically using concepts from nanophysics and engineering with Professor Ambarish Ghosh at IISc Bangalore to understand morphogenesis, and this has led to the demonstration of intra and extracellular biomaterial rheologies using magnetically maneuverable nanorobots. In the recent work led by Dr. Deban Dasgupta and Dr. Dharmapalli, they have demonstrated how these nanomotors can swim past normal cells and can adhere to the cancer-secreted matrix. To give more details on their work, we are delighted to have Dr. Deban Dasgupta, who is currently at IISc Bangalore, and Dr. Dharmapalli, currently at the University of Illinois, Chicago, to describe their work. With that, we'll also know more about their journey and things they learned when they were trainees in their labs. And so if you have any questions on their work or how to maintain a sustainable collaboration, please use the chat box option here and we'll take the question during the Q&A session. With that, I warmly welcome our panelists and our audience to Biologically Speaking, and I invite Dr. Deban Dasgupta to present their work to set the ground. Over to you, Deban.
Thank you, Shaunak. So the general idea behind this talk is nanorobotics for healthcare, and I think the idea of using nanorobotics to probe cancer microenvironment. It all started with the collaboration between Professor Ambarish Ghosh's lab with Professor Ramre's lab, and then we started seeing very new things, which eventually culminated into the project, which is a major part of my and also Dharma's PhD. And also, this is the first time nanorobotics was used to probe cancer. So let me just, So this is the general outline of the work that is being done in Professor Ambarish's lab. Most of this was done by my lab seniors. So Lakshmi, she managed to show how nanorobots can move in human blood. Before that, Arijit, another senior of the lab, actually in one of his thesis chapters showed how nanorobots may be used to measure viscosity in fluids. Shavik showed how nanoscale cargo trapping might be used to transport and release colloids and materials using nanorobots. And then the most interesting, at least from my point of view for the work that I'm going to talk about, is what Malay did in his work, where he showed how nanorobots may be maneuvered inside a living cell. And I think this showed variability in the way nanorobot moves near the nucleus versus when it's far away from the nucleus, and all of this was done in a living cell. He did manage to move it in patterns, and as you can see over here, there's an N written, so you managed to draw out all the letters of the English alphabet inside living cells. And then once the intracellular part, and probing the intracellular region of the cell was done, the next obvious question was what happens in the extracellular part of the cells, and that's where I come in, where we tried to move nanorobots through matrigel, which is an extracellular matrix, and then we saw a lot of interesting things happen. So all of this might sound like science fiction, but then I am currently, me along with Professor Ambarish and another dentist, we have started a startup which is trying to bring nanorobots into the real world. And it's called Theranautilus. It's of course not related to cancer, but it has some applications in dentistry and oral health. So just before I hand over to Professor Ramre to explain what his lab does, this is just a brief introduction to the design and structure of nanorobots. So what do we mean when we say nanorobots? And these are tiny helical structures which are about 2 to 3 microns in length and about 200 to 400 nanometers in width. And these are magnetic. So if you subject them to a rotating magnetic field, as you can see in the schematic over here, their inherent magnetic moment will follow the rotating magnetic field, and that's how they will execute the screw-like motion, which is how they will move forward or backward. And because we can control the magnetic field very effectively, we can actually command these nanorobots where to go and how to maneuver. So that's why we call them robots, because we can program and maneuver them the way we want. And we can do it with certain degree of precision. With that, I would invite Professor Ramre to explain the work from his lab.
Dharma, do you want to do the honors?
Yeah. So hi, I'm Dharma. I'm from Dr. Bhatt's lab, and so majorly, so what we try to understand at Bhatt's lab is the role of extracellular matrix and the cell surface glycans, and how these external factors play a role in cancer progression. So if you understand, we majorly work on breast cancer and ovarian cancer, and both these cancers have some of the common and unique aspects to them for the cancer progression. So some of them having their own unique architecture in the breast cancer, where the cancer epithelia in breast cancer is surrounded by are sitting on the basement membrane, just like ovarian cancer cells, and they are embedded in the milieu of the stromal extracellular matrix. Now, upon an insult, these epithelial cells undergo transformation, and then attain or lose the ability to self-restrict their growth, and then they start proliferating, and eventually they breach the basement membrane matrix and then enter into the stroma. Now, a lot of studies have been conducted to understand how the intracellular factors contribute to this transformation and then progression of these cancer cells from the primary site to the metastasis. But there are very few studies which look at the external factors such as the extracellular matrix, which is made up of bulkily by proteins and other stromal cells, and also with the other stromal cells have an influence on such transformation. So we look at, and this like extracellular matrix not just provides the structural support to the cells, but also conveys biochemical signals as well as biophysical signals. So we try to understand how these biophysical and biochemical signals come together and then drive these morphogenetic process. Over to you, Deva.
Yeah. Thank you. Yeah. So the crux of today's discussion will be nanorobotics in cancer diagnostics. So as Dharma mentioned, the key aspect of Professor Ramre's lab is to study the extracellular matrix, the extracellular environment of cancer. And we thought that nanorobots may be a very interesting tool to probe cancer microenvironment with. So before I go and talk about the results that I'm going to present here, so the initial problem that we started attacking was, can we move nanorobots to a tumor site? And I think one of the major problems that we faced was that nanorobots do not move very well in this sort of matrix. The reason being, these are very heterogeneous. They are filled with polymers. They are like a spider web. So you can think of them like a mesh of polymers, which form a spider web like geometry, which the nanorobots cannot traverse very well. So the nanorobots tend to stick in this sort of media. And that's what we struggled with for a long time. And eventually what happened was we figured out that we have to make our nanorobots very thin, at least comparable to the polymer size, so that hopefully they can move through this porous network and then actually reach the cancer cells. So this was the initial thought process. But when we did all of this, when we went back and did the nano engineering to shrink and to make our nanorobots thinner, and we started seeing motion in matrigel, which is a proxy for the extracellular matrix, and nanorobots were moving quite well in this sort of matrix because they were thinned down. We started seeing that nanomotors actually adhere near cancer cells, and this was very surprising because they were moving in the ECM, but then when they come very close to the cancer cells, they adhere to the cancer secreted matrix. This was very interesting when contrasted to the normal cells, because there was no adhesion seen near normal cells. If we increase the magnetic field, if we increase the force that the nanomotors were generating, they may eventually overcome the adhesion that they were seeing near cancer cells, but you need more force to do that sort of things. The adhesion was always seen up to 40 microns from the cell surface. So we actually had a number. We could always predict that whenever a nanomotor is somewhere close to the cancer, let's say about 40 microns from its cell surface, we would start seeing this sort of adhesion. So this was a very surprising result that we did not expect when we started this journey. And so this is a proof of what we saw. So over here on the top is MBA231 cell, which is a proxy for the cancer cells. On the bottom, there are HMLE cells, which are non-cancerous. And the bottom movie is being played at 5 times because it's much longer. It needs, you have to observe it for a much longer time to see what's happening. And you will see that the nanomotor do not adhere to the normal cells. So on the top, this guy will just come and adhere very close to the surface. but this one will keep probing the normal cell surface for a long time, and eventually it will probably get stuck to the cell surface, but it never gets stuck to the vicinity the way we see for cancer cells. So we had the chance to change the directions to keep on probing and poking the cell surface for a long time. Eventually it gets sticks to the cell surface, but yeah. And so when we did all of this, and this is the work where Dharma majorly came in with his expertise in cancer ECM and cancer biology. We saw that there is salic acid present in the cancer cell. Dharma, would you like to explain this?
Yes. So we tried to look at what could be the changes that the cancer cells are making to the ECM, when they are transformed. So what we were trying to see is, and during our discussions, we observed that this sticking could not be because of the physical barriers that arise because of the size differences or the porosity of the ECM that is secreted by the cancer cells. So interestingly, in our discussions, Professor Ambrish mentioned that it could be because of the charge, but not because of the size or the physical restriction of the nanoprobes. So one of the charge contributors in the ECM is the salic acid. So all of the ECM proteins are heavily glycosylated, and some of these glyco chains or the glycosylations are capped with a negatively charged salic acid molecule, which is a 9-carbon sugar. Now, we immediately looked at salic acids here, and salic acids in humans come in two variants, alpha-2,3 linked and alpha-2,6 linked. So when we stained the cancer cells and the immediate vicinity of cancer cells and normal cells, we observed that the cancer cells specifically had alpha-2,3 sialylation on the cell surface as well as in the immediate vicinity, where you can appreciate that the red, the entire cancer cells here are the black background is the matrigel, whereas the cancer cells are stained with salic acid, which is red, and immediate vicinity, which is also staining red, suggesting that the cancer-secreted ECM is sialylated specifically with alpha-2,3 acid. And when you look at the normal cells, you can appreciate that they have this normal growth-restricted morphology, which is very typical of the mammary epithelial cells when they are cultured on basement membrane matrix. And then the boundaries of the alpha-2,3 salic acid and the F-actin, which is cortical to these cells, coincides really nicely, suggesting that the alpha-2,3 salic acids is present only on the cell surface in terms of normal cells, while in cancer cells it is also present in the immediate extracellular vicinity. So these modifications or the sticking behavior of these nanoprobes were because of this alpha-2,3 salic acid modification, which is present in the extracellular matrix.
Yeah.
Debai?
Yes. So on the right, there is something more interesting, which is something that probably often comes out of this sort of collaborations between very diverse fields, and that's because nanorobots can possibly do this sort of things. So when you are looking at let's say the fluorescence image, you don't really see this dynamical behavior, and let me explain what's happening here. So in MDA MB231 and MCF7, which is another cancer cell, for the 231 cell, what we saw was the nanorobots sticking all around the cancer cell. So the distribution of the adhered nanorobots was more or less all around the cancer cell. But for MCF7, what was very interesting was that we would often observe that the nanorobots would just come from one side, they would be swimming and they would come from one side, they would cross the cell, maybe from over the cell. We could see the nanorobots go out of focus, and then they would go to the other side, and then they will get adhered there. Now, this sort of dynamical behavior would never show up in let's say a fluorescence image, and we would probably disregard this data, but because this was happening live under the microscope, it was very interesting for us to go back and ask what was happening to because of these two different cell types. Why is there a difference in distribution of adhered nanorobots, and what was seen? And this kind of again suggests that sialic acid is the main culprit. Is that for a 231 cell, the distribution of sialic acid is more or less uniform all around the cell, whereas MCF7 tends to generate this sort of unipolar distribution of sialic acid. So there is sialic acid only on one side of the MCF7, whereas the other side do not have the concentration of this sialic acid. And that seemed to match with what we were seeing under the microscope. So this was all falling in place when we were doing this sort of research. And now the question was, what is really happening? I mean, is it, can we shield the charge of nanorobots and then see what happens? So this is when another lab, the lab opposite to Professor Ambarish's lab, suggested that we use PFO, and this was also from some distant collaborators who suggested that use PFO, which is known to shield the charge, which is a good surface modification chemical. And when we did that, we saw that the nanomotors which were not treated with this chemical eventually got adhered to the matrix near the cancer cell. So you can see the adhered one here, which is uncoated with PFO, but the one which was coated with PFO could be seen probing the cancer cell. So this sort of proved that charge is the main culprit, that the uncharged nanomotor gets adhered to the matrix, whereas the charged one keeps on moving. So this was a nice way to tie up this finding, the effect of charge shielding. And then we moved to asking the question, what is the potential application? So what can be a real world application of this sort of science that we did? So how can this sort of things go out and help the world? And one of the very obvious questions that we thought one of the very obvious application of this would be cancer detection. So imagine these nanorobots carrying something, some fluorescent molecules, something that radiates, some signaling agent, and you move them in a tumor matrix. So if you move them back and forth for quite some time, they will eventually tend to adhere and accumulate near the cancer cells, leaving the non-cancerous cell alone. So this might be a nice way of, just by doing nothing, just by switching on the magnetic field and allowing the nanomotors to go back and forth, let them adhere near the cancer cells and leave the normal cells alone. So this is a very nice way of recognizing microtumor sites. We did this on a petri dish on our tumor model, and we also did simulations, and the numbers do match. And when we did this, so this is a very nice way we thought of detecting cancer microtumors in a tissue. The other interesting aspect of this was the numbers. When we did the numbers and the force calculation, we found something very interesting. So 231 and MCF7, these are known to be aggressive cancer cells. And we found that the force needed to unstick the propellers, unstick the nanorobots and try to move them closer to the cell surface needed more force than the ones which are not, than the cancer cells which are not aggressive. And this seems to be indicating that there might be a way to gauge the metastatic potential of cancer cells. We might be just able to do this sort of experiments on a petri dish in a clinical setup, and then say, just by measuring the force being exerted by our nanomotors, we can say whether the cell is metastatic or not, whether it's an aggressive form of cancer or not. And this was a very interesting and intriguing finding from this research. With that, I would like to conclude my talk. So we would like to acknowledge the nanobio team at IISc, which is our team, and from where all the collaborations come along. We'd also like to thank the IISc bioimaging facility and all the bodies who helped us with the grant. Yeah, thank you.
All right. Thank you very much, Devayan and Dharma. Excellent work, very interesting findings. And thank you again for describing your work so lucidly and the translational outcomes, specifically the cancer detection that really caught my attention. For audience, if you have any questions, please post it in the chat box, but I'll start with some of my own. So cancer goes through a multi-step model of oncogenesis where, depending on the stages, it secretes different types of matrix. Is it possible to generate or develop nanorobots for specific stages? Like Deba and Dharma, anyone?
I think,
Yeah.
yeah, go ahead.
I would rather prefer the people experts in biology speak for this first.
Sure,
Yeah,
anyone.
It's true that the cancer cells undergo a multi-step, morphogenetic process in the process of cancer progression, and although bulk of the matrix is being secreted by the stromal cells, for example, fibroblasts, cancer cells also secrete their own ECM during this process. Now, the question you had asked, like if they secrete different ECM at different stages, would we be able to devise specific nanoprobes? Now, I guess that depends on the composition that the composition of the ECM that these cancer cells are secreting, and the glycosylation changes or the extra post-translational modifications, which are which these ECM proteins undergo extensively. I guess if we know some of these aspects, then maybe we can try to design nanoprobes to probe, which stage, or detect the stage or the potentiality of the cancer cells. I mean, even with the current model that we have, we can kind of at least show that in terms of in the Petri dish with different metastatic potential cancer cells, we can show that the glycosylation changes in the immediate vicinity of the cancer cells is different. So this can very well be utilized to assess the stage at which these cancer cells are in.
Got it. Thank you. And I'm also trying to think from a translational point of view. You've shown that using these nanorobots you can use for microtumor detection in Petri dish. Is it possible to incorporate them in vivo, like directly into patients or mouse that you can detect it? And with that, I have a very naive question. How do you synthesize these nanorobots?
With respect to the first question, whether whether these nanoprobes can be injected into patients or mouse, I guess there was a immediate study after this one. Debayan or Professor Ambarish can answer that.
Yeah, I would prefer Professor Ambarish answer this. This is a very long project that's going on in our lab right now.
Right. Yeah. So let me take this over. So yes, indeed. So one of the main agendas that came up right after these observations were made is whether the same will hold true inside animals. And in fact, we have started this. We sort of got derailed during the COVID times, but there is an ongoing project in the lab to move the swimmers or nanoprobes inside animals, inside cancerous organs, and see if they preferentially stick near the cancer cells. One of the essential difficulties in doing this in vivo is to be able to see them deep into the tissue in a live animal. So we had to actually modify the technology to ensure that we could see them at least about a millimeter or so deep into the tissue. So that's been done. So the movies that you have seen so far are mostly in cell cultures or in microfluidic devices. So it's significantly easier. We can, with the picture that you're seeing now, we have a huge coil right now, and this is integrated with an in vivo imaging system. And this allows us to observe them maneuver in vivo. In fact, again, in collaboration with Ramde's lab and also another collaborator, Deepak Saini, the same department, we are right now have been able to move these things inside the peritoneal cavity of a mouse and also able to see some signatures of movement inside a mouse brain. So these experiments are ongoing as we speak, but the question that you raised, it's very interesting and would have tremendous translational impact if the same results are validated in live animals as well. I hope that answers your question.
Absolutely. No, I will be very much looking forward to those results. Okay, I'll take a few questions from the audience. This is from Deepak Korea. He had a first question on why do we call them as nano when they are micrometer scale? And in case, what is the material used for the nanorobot? Are they immunogenic or could be professional scavenger cells like macrophage or neutrophil?
I'm sure Deban can also answer this question, but let me, since I'm anyway visible, let me answer it anyway. See, the length is indeed more than a micron, and it will be unfair to call it a nanomotor in that sense. But the width is less than 250 nanometers, and actually the width makes all the difference out here. So I think it's perfectly okay to call this a nanomotor considering the width. In fact, I don't know if you noticed, Deban mentioned that we did not see any motion in matrigel as well as inside this living cell when the width was, unless the width was reduced below 250 nanometers or so. So this is the nano dimension is important and necessary for these experiments. Secondly, with respect to the materials, typically these things are made of silicon dioxide with a small magnetic element coming in the form of iron. Now, these are inorganic materials and they're quite harmless. And in fact, we have already had studies in which we have shown that these things are completely non-toxic. So there has been, even at a very high dosage, we have seen these animal studies in which no short or long-term toxicity has been observed. In order to make them give more properties, we can try to functionalize with the right chemicals. So for example, one can put pegs onto it. And typically, as people working with standard nanobiotechnology tools have shown that indeed PEG will allow greater bioavailability as well as less internalization by the neutrophils and all that. So all these capabilities exist within this very versatile technology.
That's great. Thank you. I'll take down the other science question as well from Jayesh Aher. How do you distribute this nanomotor over the surface of a cancer cell cluster? And is it possible to detect their live activity?
Maybe Deban, you can take this question. I think this can be answered sort of with the experimental details that you are very well aware of.
Yeah, so we injected the nanorobots is the simplest thing we could do. So what Dharma used to give me was a drop of matrigel with the cancer and the normal cells seeded, or just the cancer cell or just the normal cells seeded in a drop of matrigel. And I would have these nanomotors suspended in a fluid, which is mostly a DI water, or maybe to match the osmotic or whatever the balance of the matrigel we might consider using PBS. So this was suspended. The nanomotors were suspended in this sort of a fluid, and then I have to just inject it. Because it's a hydrogel, we never had the problem of having phase separations or this sort of problems. So it was very easy to do this experiment. We never scattered it on the cells. We just injected it at some point because this was a tumor model, and then we used to move it to bring it close to the cancer cells.
And is it possible to detect their activity?
Yeah, so I actually showed the videos so where they were moving, and that is inside a matrigel. So all the measurements, all the data that is published in this paper was mostly taken using just a simple microscope. So we could observe the nanomotors moving from point A to point B, and then we could see that they're getting adhered. We could increase the magnetic field to get them unadhered and get them moving again. So all of this was observed live using just a inverted microscope and a camera.
Okay, that's great. There's one question from Deblina. What is the fate of the nanomotors after its job is done, considering if it can be used in vivo system?
That's a very interesting question. So yeah, so the nanomotors right now, the way we envision it, they just stay in the system. But these are mostly silica. So they, as Professor Ambarish mentioned, that we have looked for adverse effects in all sorts of systems. We have given insane dosage and done toxicity, but they are not toxic at any levels. So there's no problem with keeping them in the system. Since this question came up, there are systems in the human body where it might be possible to bring them back. So if you flip the magnetic field, if they were going in one direction, you can just move them in the other direction, and then you can just bring them back to the same point from where they started, and you can clear the system in a sort of way to get rid of the nanomotors.
Excellent.
Perhaps I can add one sentence to this. So we have been quite seriously thinking about using these nanomotors for therapeutic applications. And in fact, Deba and I and another colleague of ours, we have a startup on dentistry. And on this, for these dental applications, we estimated a certain microgram of nanoswimmers that was necessary for the for the successful treatment. This is related to an antibacterial activity. The amount of silicon dioxide that is present in a few micrograms of these helices is actually less than half of the amount of silica that you will find in a pint of beer. So it's actually safer to have these swimmers than to have beer if you're worried about silica poisoning. So nothing to worry. However, it is indeed very easy to push them back. If you're able to move it in one way, just by changing the sense of rotation of the magnetic field, you should be able to pull them back, as Devan explained.
That's a very interesting analogy. I like that. There's a question from Jayesh. How do you change the magnetic field of the whole body?
So we're not changing the magnetic field of the whole body. I think we are So I think what this question asks is, can you flip the magnetic field, or can you design the magnetic field in a certain way? And that is certainly possible. So you see this large magnets in MRI, and their magnetic fields are not changing. They are mostly a single a constant magnetic field. They're a constant source of magnetic field. But over here, the magnetic field vector is rotating, and that is usually done by this sort of coils, which was there on the screen earlier. So this is a complex three-dimensional arrangement of electromagnetic coils which generates this field, and we call them rotating magnetic field because the vector is rotating in a circle, and that's how you move the nanomotors.
Okay, great. I think there's a lot of interest from the audience on these questions, so please feel free to post, but I'll move on to the next section. But this question directed to Ramre and Ambarish. I'd like to go a few years back when you thought, what was that question that seeded your work together, and you thought that, this is the time we both should come together and address this question to with our expertise that you guys have.
Ramre, you want to go first?
Yeah, I'll
go ahead, Ramre.
All right. Before we go there, I just wanted to add a couple of points which were very stimulating for me coming as a cell biologist and experiencing this entire effort. What was very revealing from the sort of for the fundamentals of cancer biology in this work was also that for the first time we had a very strong spatial measurement of how a cluster of cancer cells was able to alter its environment. Now, it's been mentioned ad nauseum as to the cancer cells remodel their microenvironment. You have something called a tumor microenvironment, and so on. But there were never any numbers. If you really look into, if you Google image these things, all you would get is cartoons. For the first time, we had given cancer cells a microenvironment, which is the matrigel. But what comes out after some time is a bunch of processes, one being chemical change, one being isotropic, anisotropic change, and the third being sort of the change in spatiality. So, one of the images actually, which was very striking, which Debayan, did not show today, which came out and is there buried in the paper, is a scanning electron microscopy that Debayan carried out in Sense, which was amazing. So on one level, what we saw were three different observations: one of sialic acid staining, one of scanning electron microscopic imaging, and a third of nanomotor sticking. And all of that was happening 40 microns away from the cells. Three independent observations. So the sialic acid talks about the sugars and charge. The scanning electron microscopy shows the altered material properties of what the cancer cells are doing to their environment, and the nanomotors are catching that edge very nicely. So this was, like a beautiful moment as far as science is concerned. Coincidences like this don't happen, and so in that way we were able to show that there is some extremely interesting sort of threshold phenomena happening about 40 microns, which is like about three cell diameters away from a cancer cluster. And depending on which cancer cell we are talking about, more aggressive or less aggressive, it's happening in an isotropic fashion or an anisotropic fashion, and so on. So that was something that is beautiful. Being able to describe it in numbers is something that was for me the big thing, going from merely a cancer cell biologist to somebody who's engaging with people like Debayan and Ambarish. The second sort of interesting thing that I wanted to add, which is a response to the question you asked as to how different cancer cells at different stages manage to perhaps secrete different kinds of matrices and so on. So what we have explored in this paper is on the basis of the material properties, the charge, and to an extent the density of the matrix that surrounds the cancer cells. We can go in much more specific manners using these swimmers as vehicles. And so as part of the ongoing project with Ambarish, which incidentally started again in an official manner in 2017, we are trying to try to catch cancer cells as they're swimming inside a big abdominal cavity, which is the peritoneal cavity. And for that, we are trying to use something more specific, which is a particular chemistry that may be native to cancer cells as they float around. And using that chemistry, or utilizing that our knowledge of that chemistry to catch these cancer cells as they float around. So it's an incredibly interesting and difficult project, because trying to image, oh, there you go, that's the scanning electron micrograph, where you again show that there is alteration of ECM beyond the cancer cells. And again, the scales come up very similar. I think Debayan shows 35 microns there as the alteration that is occurring. So again, we had numbers which were matching the biophysics and the biochemistry and the properties of the nanopropolis. I think that was what was very beautiful about this paper. So yes, specific as well as non-specific properties of cancers can be utilized, and nanoswimmers can be utilized in that particular manner to try and catch them within swathes and swathes of non-cancerous tissue. I think that's again where we are really at the cutting edge, being able to catch small populations of cancers within large populations of non-cancerous tissue. So that's as far as the science is concerned. As far as history is concerned, again, I have to thank again our colleague who was mentioned by Ambarish, Professor Deepak Saini, who's a remarkable systems biologist, pharmacologist, and somebody who studies signal transduction. Deepak's collaboration with Ambarish was before mine. And what they had been doing, which Debayan, I think, referred to as Malay's paper, was trying to sort of map the biophysical properties inside a cell. Coming from a background where, for me, the tumor microenvironment was of much more interest. The extracellular changes by a tumor and around the tumor was of much interest. I naturally sort of joined and jumped into the bandwagon by asking Ambarish, what if we can similarly map the biophysical properties around cancers as a way of catching them. So this was this naive sort of idea with which we launched the idea. The other sort of interesting point was, can we also work with ovarian cancer, which is a floating cancer where you don't have to use these propellers to move through all kinds of more difficult environments. You can directly sort of fluidically reach there. But in the process, as happens, we jumped from problem to problem till we came across something that was hard enough, but also interesting enough to solve. And with students like Dharma and Debayan, we were able to do so in a remarkably short time frame. So that's as far as my sort of entry here is concerned. Ambarish, you might want to add.
Yeah, I think you covered most of it. See, so I have been trained as a physicist, and to me, the whole world is a sphere, and there are no complexities. Well, there are complexities, but we approach it differently. So when we started, really working on these tiny swimmers, we of course realized that while they are, it's very interesting to observe their motion in model fluids, and you can make all sorts of very interesting physical measurements like viscosity, elasticity, stuff like that, like mechanical measurements. We realized that the real power of this system lies when you use them in sort of really complex and heterogeneous environments. And, even something, this was a really old project, I think back in 2013-14. All that we did was to simply take the swimmers, take it out from water, and instead putting it in blood. And we could not move them in blood. This was just some ex vivo, just some blood that was donated by somebody. We could not move it, and we realized that, and this has to do with the corrosive nature of blood, the protein corona formation, and all of that. So we realized, of course, that was a failure, at least for many months, till this very bright student figured out how to circumvent this issue. But it also taught us that this is a unique opportunity, because now you have a swimmer, now you have you have a probe that will allow you to really look at a sub-micron scale small heterogeneities. And initially, and this is what Ramray just told you, with Deepak's, this collaboration with Deepak, we realized that the inside of the cell is an extremely heterogeneous mechanical environment. In fact, I'll take a step forward, and I would say that close to the nucleus, the elasticity increases dramatically. And we sort of made some measurements on that. When we started this work on ECM, of course, in addition to biophysical measurements, we also wanted to see, well, what would it take to move these swimmers in a heart tumor, in a cancerous organ? So that's how this project all got started. At this time, we had absolutely no intention of looking at charge whatsoever. So this was a complete surprise. And, it's just over the course of a year or so in which this experiment started, the constant brainstorming that happened between our groups, we realized that, well, not only can these things measure mechanical properties like viscosity, elasticity, local porosity, but it can also measure charge. And charge is such an important parameter in cancer biology. I had no idea. And this is really what Dharma and Ramray taught us. And I must take a step forward. This sort of inspired us to say that, okay, can these nanoswimmers now also measure some other types of heterogeneities? Why does it need to be biophysical heterogeneities? Can it, for example, measure biochemical heterogeneities, like, for example, the sialic acid? Now, can we detect this chemical species using some other way? So this is where sort of other physical techniques come in that we are working on. For example, one of the things we are trying to do right now is to try to do local NMR type measurements and see if we can detect local chemicals. So not only this collaboration, it started somewhere, it has led to this very interesting observation, which of course we are pursuing, but also inspired us to mold our research questions and future directions in a way. So it has been a fantastic learning experience, at least for me, and definitely, everybody in our group. And we really look forward to having more projects along this.
No, thank you. Yeah, thank you very much, Operation Ramray, for beautifully explaining the history behind this collaboration. You've actually answered a lot of my other questions that I was about to ask here, especially the thoughts on the training and development opportunities that we learn by doing this collaborative works. I'll have a question for Debayan and Dharma. Amrish has already explained a few of the Eureka moments, but if you both can share a few of the challenges that you have faced while doing this project. So it's easy for the PhD students in our audience or those who are aspiring PhD students, they can actually feel what goes, the challenges that they face during the grad school.
I'll start first.
Sure.
So yeah, like both Ambrish and Ramray had mentioned, the project started somewhere, and then it now the way it headed and the directions it took was completely unexpected, and the discovery of the charge, the presence of the charge, and the how charge affected the sticking behavior was completely totally new to me. And coming from a biological background, I may not have, or I wouldn't have recognized that the sticking behavior of these nanopropellers is because of the charge. I mean, I would have immediately thought that it could be because of the porosity that is coming from the ECM secretion that is being made by the cancer cells. So the collaboration that this collaboration had taught me that it's sometimes we may have to think out of the box to understand some of the biological questions. And coming to the challenges, so in terms of technicalities, the biggest challenge I felt was imaging. So the scales at which the cells and the nanopropellers were, and the kind of imaging that we were trying to do using confocal microscopy, it was a bit difficult as to get both these entities in one physical space, as to the microscale and the nanoscale. So that was a bit of a challenging, and the depth of the scaffolds that we were using was also a bit of a challenging in terms of technicalities that we were trying to do. But we have come up with a lot of, after a lot of brainstorming with Debayan and Professor Ambrish, we could resolve these and get the beautiful images that Debayan had presented in this talk.
Amazing. Debayan, do you want to add something?
Yeah, I think Dharma spoke about a lot of the scientific challenges and how we overcame most of them. And those are true from my point of view too. But so there's something interesting which I think both me and Dharma faced, and what he was talking about is when you're doing this sort of experiments, and mostly in interdisciplinary experiments like this, there is no precedent. I mean, you cannot look up. So there are certain experiments which you can just look up the methods, modify them, and do new things. But in this sort of, at least for nanoscience and this sort of interdisciplinary subjects, there is very rarely any sort of precedent to do, and then you keep thinking, how do I go forward from here? And I think that's when, I mean, having good collaborators is very important, because I mean, they have a very different way of looking at things and very different perspective on solving these problems. And when you hear their point of view, it becomes very interesting, and you can come up with new methods of solving this sort of conundrum when you face them. And I mean, I have seen that firsthand happen with me. As Ramre was mentioning about the SEM images, and because my training is in nanoscience, and I am programmed to go and look for an electron microscope whenever something goes wrong, and this was something new from Dharma's point of view, and so I think that sort of gave us a clear picture of what's happening, and that this is the kind of thinking that often evolves from having a very interdisciplinary work going on.
Very well said. Well, I think we are running short of time, so I'd end by one last question to Ramre and Ambarish. This is mostly your suggestions to young investigators in our audience. Is how do you choose collaborator? How do you manage your time with your lab's own ongoing work, and you want to collaborate with someone else? And this also brings one of the questions from the audience is, how were you all able to cooperate well as team to accomplish this worthwhile goal? And if you can end by saying how do you discuss authorship when it comes to publication?
Okay, maybe I'll go first.
Sure.
I'll keep it short. So, I just want to add one important thing that I have recognized in collaborations. Well, the way I at least go for it is, I generally go for collaborations. Not all collaborations work, because there has to be a match on many different levels. It has to be a match on what is interesting science to each of us, what is insightful, and when is it the right time for it to be insightful and deep enough for both collaborators, and of course a certain temperamental match, a certain attitudinal match is very important as well. The other and more important point, I would say, is that as far as collaborations are concerned, what I've realized is it cannot work again in terms of silos where I trust your work and you trust my work. I have to understand what you're doing, and I think the other collaborator has to understand what I'm doing, to the extent that they criticize our inputs, our biological inputs, and we can try to criticize their physics inputs. So it has to work not just at the level of trust, but a certain level of healthy skepticism as well about what is evolving, and constantly questioning that, and actually spending time. Dharma, I know, we've met every week, every second week for the last 6 years, and that's the way we work. That's what a collaboration is. It cannot work in parallel. It has to work in series. It has to work together. And it has to work exactly in tandem, sitting together, bringing students together, and breaking our head and disagreeing every day. That's, I think, crucial to collaboration, as far as or whatever cooperation was mentioned. And also, a last point: a certain level of craziness mixed with a certain level of discipline is very crucial to collaboration. I think both Dharma and Debayan in this case brought in a certain level of nuttiness, but they are both very disciplined workers, in that sense, scientists, right, at the moment. And that discipline, I think, and is what resulted in a terrific paper from what could have been a good paper. So I think I'll rest my case.
Excellent.
Yeah, I think Ramre pretty much covered most of the things that I also wanted to say. So perhaps I'll say a few additional things. See, there can be types of collaboration where somebody maybe makes something and hands it over to the next person, like a serial type of stuff, in which as long as it has some level of maturity and what's coming in is perfect, things get started, things works. But at least the type of problems we are doing here, where we are trying to use we are in uncharted territories. That's what Devan was mentioning. There are no rules of the game. So we got to work with each other, question each other, talk to each other. And, when we had these group meetings, typically we keep our egos out of the room and, completely ask questions like, I am known to ask, I don't know how many biology questions to Ramre in every one of our weekly meetings. And, I don't know, at least I now know, I know how to understand what he's saying. I know his language now. And that's very, very important. You had also asked about authorship. So I think this is something where, very frankly, in this particular case, it was very obvious because people really worked together and have put tremendous amount of both efforts as well as thinking in both of them. So I think we sort of look at it on a case-by-case basis, depending on the amount of intellectual as well as, inputs, different types of inputs that came into the project. So I don't think there is any golden rule in this. The part that probably the main reason why it really, the whole project became really successful, and I think that's something that I feel many such multidisciplinary collaborators should think about, is we decided to write the paper only after the questions were answered. And the question was very clear to us. That observation was, why are they getting stuck? And we did not stop till the question was answered. And only at that time, we decided that, well, let's now write the paper. So we had a complete agreement on that part, and which I think is the main reason why this collaboration was very successful. And that's also probably where that temperament matching is very important among the group members. So these are the few things I wanted to add to what Amri told already.
Amazing. Thank you very much, Ramre and Ambarish, for sharing your insights on successful collaborations. Thanks again to Deban and Dharma for beautifully explaining your work. And once again, thanks to all of our panelists for taking the time to participate in this discussion. I'm greatly benefited. I hope all of our audience are also greatly benefited from these collaborations. And thanks to our audience for asking these phenomenal questions. Really enjoyed. And before I stop, I request Mohit to share his last few remarks before we close today's session.
Thank you, Shanak. Thank you so much, Ramre, Ambarish, Dharma, and Devan. I have learned a whole lot in the past 1 hour, both about the science as well as about the nuts and bolts of collaboration and what amount of perseverance and, as Ramre said, a good mix of craziness and discipline it takes in a sustained manner to get to this stage. So thank you so much for sharing that. And that's the whole point behind this series, that can we bring all the trainees also together and have this discussion, what goes on behind the scenes. And as Ambarish was saying, there can be these more service type collaborations, but then there can be more deeply engaging collaborations, of which, of course, your work is a remarkable example. So thank you so much for sharing this, and all the best for your future work in vivo and translational aspects of it. I'm really looking forward to seeing more exciting things coming from both of your groups. Thank you once again, and thank you everyone for joining.
Thanks, Mohit. Thank you, everyone. Please stay tuned and follow us on social media platforms to get updates of our future sessions. Thank you, everyone, and have a great rest of your day or evening, wherever you're listening from. Thank you very much.
Thank you. Bye.
Bye.